I've got the pleasure of introducing our speaker. And so our speaker grew up here in Portland. She graduated from Wilson High School. And she just finished her freshman year at Stanford University, where she's studying mathematical computational sciences there. Okay, so she's going back in a few weeks, and it's really been great to have Jorden here during the past few months. One of the group I work with a group, that does sort of artificial intelligence and deep learning for diagnosis of eye disease, and it's been really, really nice to have Jorden around. She's smart and she works hard, and she's done a lot of neat things this summer, and so that's what she'll be talking about. And so if you can help me welcome Jorden Schuster.
Dr. Chang. Okay, I'll move this. Is that better? Okay. Okay, so as Dr. Chang said, one thing that we've been doing down in his lab at Casey is deep learning for diagnosing retinopathy prematurity. And along with retinopathy prematurity, one big impact is image quality. So Aaron Coiner, a PhD in our lab, recently made a deep learning-based method for looking at image quality. And so what I was doing was looking at the relationship between image quality and diagnostic ability. So the first step in looking at this and looking at what is ROP, and ROP is retinopathy prematurity, and specifically what plus disease is, and then what the automated systems do, and that being the diagnostic system and image quality assessment system, and then the relationship between ROP and the diagnostic ability and image quality, and then other possible measures of quality. So retinopathy prematurity is the leading cause of blindness in children. So with more neonatal care available, there's more premature babies born. And at about 16 weeks, your retinal vessels will start to grow, and by a full-term pregnancy, you'll have retinal vessels that grow to the edge of the retina. When babies are born prematurely, they have the opportunity to grow abnormally and cause bleeding or scar tissue, which in severe cases can pull on the retina and cause a retinal detachment. So it's completely treatable if you catch it early enough, but that's the trick. You have to catch it early enough. When to treat it is when you see plus disease. So here you can see a few images. Plus disease on the left here is defined by tortuosity and dilation in 2 or more quadrants. So it's kind of difficult to see, but here you can see kind of angry-looking almost vessels that are squiggly and a little bit thicker. Pre-plus disease, it's harder to see because of the quality on the screen, but still has tortuous vessels, but a lesser amount. And then normal has very thin, not dilated, straight vessels. So in kind of the most severe cases, it looks like this, which is very easy to classify as plus disease, but where you have tortuosity and dilation, and it's very easy to see it's in even more than 2 quadrants of the eye. But the majority of the time, it's a lot more ambiguous. So here, there's some subjectivity and qualitative parts of diagnosing plus disease rather than quantitative. So here we have 2 images that are both pre-plus, but one is on the lower end closer to normal, and one is on the higher end closer to plus. So in this particular case, they were taken from a set of 100 images, which 8 raters individually graded each image, normal, pre-plus, and plus. With both of these, it was not a complete consensus. So in both of these, yeah, that's better. In both of these, on the right here, the closer to normal one, about half the people said it was normal, and half the people said it was pre-plus. And then on the right, about half the people said it was plus, and half the people said it was pre-plus. So in these cases, the right answer is pre-plus, but the diagnosis may be variable, even among the best experts. So Dr. Chang did a study at one point looking at inter-rater agreement more specifically. And this was on the same, or 34 images, 22 experts, and only 4 images out of the 34, all 22 experts agreed on the same 3-class classification. So there were 4 images that were looked at like these where every single expert said the same thing, which is very low. And so it just shows that diagnosing ROP can be very imperfect. Plus disease can be imperfect. And so this is the motivation for computer-based analysis, to give a very objective, only looking at what's there and not any gut feeling where this looks worse, this looks less worse, not as bad. So James Brown, previously at Mass Gen, created a deep learning system for this. And to explain a little bit what deep learning is, they used a convolutional neural net, which at the simplest takes in an image and outputs probabilities. So it takes in an image, and each layer shown here, neural nets are made up of layers, does a different algorithm or process on the image to the point where it gets to the end, and at that point you can make a decision based on probabilities. So with machine learning, you have to have training and testing. So in a large data set of around 5,000 images, the system was trained and on labeled data. And one thing that's really hard about machine learning in any medical field is labeling data accurately. So in this case, the best you can possibly do is to get a group of experts to come together and label data. And so one problem is that we kind of showed previously that experts don't always agree. And so that is the case when making an RSD or reference standard. And so it's very difficult and time-consuming to come and have the best experts look at images, 5,000 images, and say, this is what I think it is. And then if there's disagreements coming through and making a consensus on what we think it is. And so that's a very large component on how a machine learning system learns, is going through the process and guessing on what an image is and then making a label for it and adjusting what it thinks it is. So in our case, we start with a fundus image, which is this image of the retina here. And then there is 2 steps in making a convolutional neural net. The first of which learns and has to make a segmentation. And so this is what we refer to as a segmentation. It pulls out the vessels. And so one thing that this does is it takes away the coloration of the image. And when you're looking at plus disease, you're very specifically looking at the vasculature. So is it dilated? Is it tortuous? And by pulling out the vessels, we can be sure that it's only looking at what we think it's looking at. And so from this, it takes a classification type machine learning algorithm, which is the other end of the convolutional neural net, and outputs 3 probabilities. And the 3 probabilities correspond to the probability of the image being a normal image, it being a pre-plus image, and a plus image. So here you can see this image has dilated and tortuous vessels, and that carries into the segmentation, which you can see a little bit clearer. And then the probability of normal and pre-plus are almost 0, and the probability of plus is almost 1. And so one thing you can note with when it outputs these probabilities, they always add to 1. And so in some cases, it can be really sure, like this image, and then in some cases, it can be really unsure, or that may be a case where 2 of the probabilities were close to a half, or in very, very unlikely cases, they're almost evenly split. So Aaron Coyner's algorithm, which was an automated assessment of image quality, was made basically so if you're looking as an expert at a retinal image, and it looks like the one here where you can barely see anything, how are you supposed to make a diagnosis off of it? And that carries into how's the computer supposed to make a diagnosis off of it if we can't see anything? And so originally, the idea was to see is the computer doing better off of better quality images? So we can't expect it really to do well on poor quality images. So in the original test that I was talking about just now that James Brown made, he actually took out most of the non-acceptable quality images like this. But in this case, we want to look at all qualities of images, and so we get an image and a probability, just like the last one. Since it's convolutional neural net, you take in an image and you get a probability. And close to 0 is poor quality, and close to 1 is high quality. So you can see going across the side here, this very poor quality gets almost 0. The medium quality, where you can see some vasculature on the outside, gets about 50%, and then the very good image gets almost 1. And so looking at the relationship between diagnostic ability and image quality, first we wanted to make a test set. And what was important for us was that none of the previous algorithms had trained on the data set that we had used. So for those who are not as familiar with machine learning, if you feed an algorithm a image that has trained on, it will have memorized it essentially. So you would get artificially heightened performance if you were to give it data that it has trained on. So what we did was taking a data set of 6,000 and removing all the images that were trained on, and so we were left with 1,122. And then deciding what measures to calculate the performance on. So we used AUC being area under the curve, ROC curve, sensitivity, specificity, NPV, and PPV. One thing that's really important about medical uses of machine learning in particular is to use more holistic measures of what good performance is. Because if you have a disease prevalence that's very low and your machine learning algorithm maybe does a 95% accuracy, and you have maybe a 3% prevalence, then it could have missed all the pertinent images. So we wanted to make sure that it's missing the images, that it's getting right the images that we want it to get right, and it's missing the images that maybe we don't know why, but it's getting the correct ones right and wrong. And then we looked at interpreting the results. So the graphs here are not completely intuitive, but the x-axis is image quality cutoff, which is kind of hard to see, but looking at different image quality cutoffs, and at each one taking out all of the data that is below the cutoff. So at 0 on the end here, you are using all the data because all of the image quality values, and these being the probabilities outputted from the previous image quality CNN. All the quality values that were above a certain point, and then running the numbers on them. So on the end here, that's the entire data set because everything is above 0. And then when it gets closer to 1, you have only the very best quality images that you're running these diagnoses on. Another thing to note is that there's 2 graphs here. So the output probabilities from the diagnostic CNN is 3. We have 3 probabilities. And for most of these measures, it only requires 2, or you can only really give it 2. So we broke it down into normal and pre-plus and plus together. And so that would be more of like a screening situation, and then A plus and versus pre-plus and normal. And so kind of what we expected to see was a curve more like this, where it increases as quality increases. But we can see it's relatively flat. And if anything, it almost curves down as quality increases, which doesn't totally make sense, since for us, when we see a better quality images, it's easier to make a diagnosis off of. So that may not be the case for computers, in this case in particular. But 2 things we were thinking from this. One is, does image quality matter, really? Is there a difference, or does the image quality algorithm work? So after going through and looking at the image quality algorithm, we kind of concluded that, yeah, that's a high-quality image that it said was a high-quality image. That's low. Maybe it's working fine. So we were kind of left with the idea that quality isn't positively correlated with increased performance. So it's more, it does the same across any kinds of qualities. And then after seeing this, since we had hoped for a more like increasing type curve, we were looking at other different possible types of quality. And so the first of which being the proportion of images segmented. So these are 2 examples of segmentations we saw when we were looking at the way the CNN works. It makes an image and then a segmentation and then 3 probabilities based off of it. So one thing that's different than this than regular image quality, like looking at the haziness of the image, is that the computer is seeing the segmented image. So the quality of what the computer is seeing is more based on the segmented image than the image quality of the haziness, because the computer is actually much better at looking at this, like picking out vessels and segmenting them than people are. So here we think that a higher quality image would have more segmentation. So we can see at the top here, we have a pretty high quality image, and we see a lot of vasculature. And then at the bottom, we have a very low quality image. So what happened here is in very, very low quality image, the CNN outputs a grayscale image, so a matrix of intensity values. And when the grayscale image is almost completely black, the intensity values are very low. And so the CNN that classifies and puts out the probabilities looks at a binarized image, meaning it's no longer grayscale, it's all 1s and 0s. So in between when it segments and when the probabilities are being made, it is binarized. And so when it's almost black in the grayscale image, there are usually very small pixels that have really small intensity values. So here what happened was when it's thresholding, so it will pick a threshold and then binarize above the threshold. Since the values are very low, the threshold is very low, then it happens to segment things that we don't really want. And so in this case, that's a way that this wouldn't necessarily work because it's segmenting things that we don't really want to see. But in the majority of cases, with a higher quality image, you see more segmented. And so this is the graph in the middle is image quality scores versus the proportion of the image segmented. So they're relatively loosely correlated positively. And so any background noise is picked up, which is kind of a challenge here. And it automatically counts it as relevant vasculature. And then this is keeping track of the quality of the segmentation rather than the image. So it's not necessarily supposed to be exactly like the quality measure that we have before. So the same graphs were run on these ones, and unfortunately they're definitely worse looking. So it's not necessarily clear to us why they may decrease so much as the proportion of the image segmented goes up. But you can see it's kind of steady and then drops off. So the proportion of the image segmented just has too many things different than what we're really looking for. And so it's not necessarily a good quality measure. The second one was the top 5 contiguous components. This being like, if you were to look at the top image, a lot of the vessels are connected, and so they're very long and take up more of the image. So it would take the top 5 largest pixels, like groups of pixels, the groups being parts that are connected. So that would eliminate the fact that it's picking up on all of the spottiness of this image. It might only pick up on the 3 largest or 5 largest spots. And so in the high quality images, this ends up being almost exactly the same as the number of pixels segmented and doesn't really change anything as a quality measure compared to the last one. In some cases that are very low quality images, instead of like long skinny lines like we would hope, you get more like blobs in the middle, which unfortunately count as many pixels maybe as a long skinny line, which you can't control as much. And then this is the graphs kind of for those. And they look almost identical to the total number of pixels segmented because they're very similar. But you can see it decreases as quality decreases, or as the number of pixels increase, which is not really clear. We saw in the original that in some cases it just, on total number of images, there were more higher quality images missed than lower quality images, which we're in the process of looking at why that happens now. But in this case, it decreases by a much larger amount. So in general, what we came up with was at the state that deepROP is now, which is a diagnostic system, is it's not clear that there's a correlation between image quality and diagnostic ability. So as the state it is right now, that has the same performance on lower quality images and higher quality images, and that may change in future ones where we would maybe something changes with the algorithm itself, and it now diagnoses more higher quality images, but with the way it is now, it's unclear on if there's really a correlation between them. And then in a lot of machine learning applications, what's really published and publicized is where it gets things right. It had a 98% accuracy, but in our case especially, when the stakes are high, you need to diagnose plus disease for a child to get treatment. We need to look at where images fail. So what we're doing now is going through, we think that you can classify errors from this diagnostic system into discrete bins, and then maybe that would tell us something about how to change it in the future. For example, if we go through and it looks like all the cases it missed were borderline cases, that may argue for a more continuous scale, since, as we saw earlier, things may not perfectly fit into 3 bins. They may be closer to normal than pre-plus, or closer to plus than pre-plus, and it's hard to put it in between. Or the segmentation errors, and maybe change something about the way that the algorithm segments, or it's quality issues, in which case you set a cutoff value. So that's mainly, now Missy and I are working towards that and going through all of the images that it missed and looking at why it got it wrong. Could there be underlying reason that a lot of them are getting it wrong? And yeah. So I just want to thank Dr. Chang and Pete Campbell, who was not here, and Susan and Misty and Aaron. We're all big people who were always in the KCI Institute lab and DMICE in the National Library of Medicine. But mainly, one thing I did not expect from informatics, I expected to be mostly at a computer, but the group down there makes a very good point of bringing people together for meetings and conferences with other people in the country and staying up to date on what everyone else is doing and being very supportive and checking up and helping you get things that you need, which I very appreciated. It was a very good time working there. So, yeah, that's all I have.
Thank you for that. I just had a quick question about you said that the quality, wasn't correlated with the performance, but I wondered whether that was, the correlations were kind of equally true, whether the performance meant agreeing that something was negative versus agreeing that something was positive. Was there any difference in how it performed with the images that it was going to find to be negative versus how it performed with the images that were positive?
So one issue that we had with our data set of, because the prevalence of plus disease is lower, in our data set of like 1,100, we only had like 39 plus images. And so it didn't seem like there was a big connection between the positive cases getting wrong and the negative cases getting wrong. One thing that was interesting that we tried to go about fixing but couldn't figure out a way to do because the data set was so small is that rebalancing the distribution of the quality of images. So kind of unfortunately for us, a big majority of the plus disease images, I think 11 out of 39 were poor quality, and what the quality assessment measure called poor quality. And so we tried to go about finding other images, but of course the data sets are what you have, and you can't, to prevent data leakage, being you trained on an image before, we did what we had. So it didn't look like there was a difference in positive versus negative cases, but of course we had a very small amount. So. Mm-hmm. Yes.
Okay, while they're switching out, I'll introduce the next speaker. So I'm Dave Dorr, and I'm pleased to introduce Nidhi Pai, who is a senior at Oregon State studying public health and human development and family sciences, and she really did a wonderful job. I give our group, Care Management Plus, studies basically how data, information, and knowledge can impact the health and well-being of vulnerable populations, and you'll see she took on a really important aspect of that with this study by looking at psychosocial factors on patients with multimorbidity.
Oh, I turned off the monitor. Sneak peek. All right. My monitor keeps turning off, sorry. Monica, I'm not sure what happened. Thank you. Perfect. All right. Can you hear me? Yes? Cool. All right. Thank you for the introduction, Dr. Dorr, and thank you everyone for being here today or tuning into the live stream. I know a couple of people I know are. I had the privilege of being the Care Management Plus intern this summer and working on several grants with the Care Management Plus team. And I'm going to be presenting on one of those grants, which centered around analyzing the effects of psychosocial factors on patients with multimorbidity. So quickly about Care Management Plus, the CMP team basically has a mission to better understand how data, information, and knowledge can be used and leveraged to shape and transform health outcomes for vulnerable populations. So coming in from a public health major with a strong foundation of public health knowledge, but no experience in informatics or what that really meant, I learned a lot this summer in the last 10 weeks from the CMP team about how public health and informatics come together to shape health outcomes for individuals and populations. So I know we have an audience today with various backgrounds in public health and informatics. So before I jump into the research, I just want to start by painting a quick picture of the healthcare challenges that are driving the needs for research like this in the first place. So starting with models of healthcare, which are basically ways of conceptualizing health or thinking about health, achieving health. So our current U.S. healthcare system is largely based on what is known as the biomedical model of health. In this model, the primary focus is on the biological and the physical aspects of disease and illness. And some of the primary aims for patient care are to diagnose, treat, and cure so that patients return to a pre-illness state. So there are a lot of strengths with this model, and it's brought a lot of advancements in life expectancy and the quality of life and patient care that we can provide. But one of the significant gaps in this model is that the emphasis on biological determinants means that social and behavioral determinants of health are generally excluded or overlooked in care. So now there's a growing body of understanding on social and environmental factors that shape our health and need to be addressed to achieve holistic health and well-being. And these are known as the social determinants of health. So as you can see in this table, there are a lot of examples of social determinants of health, everything from education to food security to the quality of our healthcare system, all of which interact to shape the health outcomes highlighted there in the orange bar. So the last model I'm going to discuss today is the biopsychosocial model of disease and illness. So the BPS model looks at biological, psychological, and social needs of patients and how they all interact to shape disease and health outcomes. And the BPS grant was the foundation of the grant that, or not the BPS grant, the BPS model was the foundation of the grant that I got to assist with this summer. So despite a growing body of knowledge on social determinants of health and the impact that they have on our health and well-being, there are still significant gaps in integrating this knowledge into clinical care and into workflows. And a significant way that this manifests is in EHR documentation. So you've all probably heard of vital signs, which are they're basically measurements that are indicative of various aspects of our health. So we have biological vital signs, things like blood pressure, height, weight, BMI, all of which typically address our biological and physical needs. These biological vital signs are entered into structured data fields in EHRs like Epic, which makes it easier to evaluate and act on them. Then we have psychosocial vital signs, which are factors like financial resource strain, housing, loneliness, stress, all of which are indicative of psychological and social needs of patients. Currently, what we're seeing is that these psychosocial vital signs are generally entered into unstructured data fields like notes, which makes it a lot harder to evaluate and act on them consistently. So now what we're seeing is that we have a lot of unstructured notes, but that this is a largely untapped resource of a lot of rich information that could have a lot of implications for patient health. Specifically for psychosocial factors, there's a lot we still don't know about how psychosocial factors interact with biomedical determinants of health, how they
how and if they exacerbate disease and if they put certain populations at higher risk of adverse health outcomes. So that leads me to an overarching question of this research, and that is, can patients with psychosocial factors be identified despite this unstructured documentation? And the answer is yes. Past work by Greenwald and team used keywords and natural language processing methods to mine several thousand patient notes for physical, cognitive, and psychosocial constructs. The following year, a study by Dr. Bejan and team used various methodologies to mine several million patient notes for homelessness and adverse childhood experiences. So this, Dr. Bejan's work was the foundation of the grant that I got to work on this summer. Our grant looked at 4 psychosocial concepts. One of them was homelessness, which was replicated from the previous grant. And the other 3 concepts we looked at were chronic stress, social isolation, and financial insecurity, all of which are known to be associated with adverse health outcomes of patients. So the specific target population that we looked at in this study was adults ages 65 and older experiencing multimorbidity, which is the presence of 2 or more co-occurring chronic conditions. So currently, this is based on recent data, about 1/4 of the American population is experiencing multimorbidity, and over 75% of people ages 65 and over are experiencing multimorbidity. And despite advancements in healthcare and medicine, this rate is increasing. I don't know why that's right over the words. What we also know is that patients who are experiencing multimorbidity are more vulnerable to a variety of adverse health outcomes. So for example, patients with experiencing 5 or more chronic conditions are found to experience 5 times the rate of risk of functional loss and 90 times the risk of hospitalizations, and also represent a disproportionately high percentage of healthcare costs. So this is what shaped the inclusion criteria for our study. To be included in the analyses, patients had to be 65 years or older by the year of analysis, had to be experiencing multimorbidity, and the third inclusion criteria was that they had to have a minimum of 1 identified seed word. So seed words are basically single or multi-word expressions that are determined to be associated with the concepts of interest. Now, why is this important? Well, each of these social determinants is very complex, and there's a lot of indicators that a patient could be experiencing them. For example, if a patient is found to be residing in a shelter, could be an indicator that they're experiencing homelessness. If a patient is repeatedly referred to a stress management class, could be an indicator that they're experiencing chronic stress. So basically, collectively, these seed words, these are the ones we determined for our research. These seed words create a phenotypic profile for each of the social determinants that allow us to broaden the scope of the search and identify even more patients who might be experiencing these psychosocial constructs. So before we get to the point of identifying seeds excuse me, in our methodology, this research started with a data set of 3.28 million patients from 120 different sites. This data set was then narrowed down to just 2016 and 2017 data containing information on 80,748 patients who met the inclusion criteria. Following this process, we identified notes of patients using seeds who were found to be experiencing those psychosocial constructs. Any seeds that had identified notes incorrectly or erroneously were either reformulated or removed from the analyses using negation filtering. Following this process, based on our seed profile and how well all of the patient notes matched that seed profile, patients were assigned a relevance score, with higher relevance scores indicating better matches to those seeds and probably higher prevalence of that concept in their notes. So this 4th stage is where I actually joined the CMP team this summer, the manual annotation process. And the point of this phase was basically to validate the retrieved list of patients and to make sure that the patients that had been retrieved by the software were actually experiencing the psychosocial constructs that they said they were. And so for this process, a colleague and I went through 400 patient notes in total. And this process basically involved reviewing patient notes and assigning patients a score on a 0 to 5 scale based on how well patient notes matched and how well, sorry, I lost my train of thought, based on how well the psychosocial construct was present in their notes. So a score of 0 indicated that the psychosocial construct was definitely not present. Score of 5 indicated that it was definitely present. And so if a patient had a score of 3, 4, or 5, that was deemed a true positive for our analyses. So these are the results from that manual annotation of 400 patients, which revealed pretty moderately to highly significant precision ranges and average scores for patients, with precision ranges going from 0.78 to 0.93 and average scores ranging from 3.93 to 4.65, which indicated that most of the patients that had been extracted were indeed true positives for the psychosocial construct. So after this phase, the grant was submitted pretty shortly after this phase. So in the last few weeks of my internship, I got to spend my time learning about R and statistics, reteaching myself statistics, and exploring the data to see if it could answer some of the questions that we had. So my main questions with this data were to look at the effect of psychosocial factors like the 4 I mentioned earlier, and to see if they had an effect on exacerbating or increasing the frequency of health outcomes for patients with multimorbidity. So the specific health outcomes that we looked at for this data were unplanned healthcare utilization and disease exacerbation. To look at the impact on these outcomes over time, we used the 2016 and 2017 data set of 80,000 patients, and then compared it to outcome data from 2018. So for this outcome data, we used 2018 information on hospital and emergency department visits by that data set of patients. And for disease exacerbation, used ICD-10 diagnostic codes to assess the presence of 19 standardized mental and somatic health conditions in patient records. So just a quick look at what that data actually showed. So of our total population of 80,748 patients in that data set, about 55% were female, 45% were male, with an average age of about 71.98, we'll just say 72 years. Of the 4 psychosocial factors, the one that seemed to have the highest prevalence was chronic stress, with about 13% of the 80,000 patients identified as having a true positive for chronic stress. On the other hand, social isolation seemed to have the least prevalence of the 4 here, with just 0.48%, but still 362 patients identified as having social isolation. And lastly, about 4,000 patients were identified as having 1 or more hospitalization in the year 2018, indicating a rate of about 50 per 100, not 100, 50 per 1,000 patient years. So the predictive analysis method that I used in R this summer was primarily binary logistic regression, which is a method that basically looks at the relationship between a dependent binary variable and a set of independent variables. Basically what the regression does is it predicts the probability that a certain binary outcome will occur based on a set of predictor factors. So my first question in this analysis was to see if there was a relationship between the presence or absence of a psychosocial factor and the probability of a hospitalization. Either yes, they were hospitalized, or no, they did not have 1 or more hospitalization. So the results on this, the results on this showed that odds ratios were greater than 2 for all 4 psychosocial factors and had statistically significant p-values. And this indicated that yes, indeed, for all 4 psychosocial factors, if a patient had one of those factors present in their notes, they were at an increased likelihood of hospitalization if they were patients with multimorbidity. And my second question was similar and looked at the patient relevance scores that were determined based on how well patients matched with the seed word profiles we'd created, and then compared that to the probability of hospitalization. So I was able to generate these graphs based on that regression. And it looks like for chronic stress, homelessness, and financial insecurity, an increase in relevance does indicate an increase in the probability of hospital admission. Now, the exception to this in this example was social isolation, which did not appear to be statistically significant in this model. One reason for this could just be because of the volume of the data that we had on social isolation. We had significantly less than on the other psychosocial factors, which could have affected the probability of social isolation and hospitalization. But regardless, I think all 4 of these graphs still show us a really interesting relationship between these 2 factors. Now, my third research question was to look at the relationship between the disease diagnoses that we had extracted and the presence and the absence of a psychosocial concept. So to see if having a certain disease diagnosis increased the likelihood of having a psychosocial factor as well. So to start with this, I started by running a multivariate analysis, where all 19 of the diagnoses we were looking at were compared to each psychosocial factor individually. And this is where I ran into a fun challenge with logistic regression. So in this example, with chronic stress, there are several diseases that are statistically significant predictors of chronic stress. One of the conditions that is not is substance use. So to explore this a little bit more, I ran a univariate analysis, which just compared substance use to chronic stress. And in this example, you can see that substance use is now a highly significant predictor. So when I first saw this, I was really confused, thought I had just messed everything up. But turns out the challenge has a name, and it is multicollinearity. So that is basically a phenomenon in which 2 or more independent variables are highly linearly correlated. And so this is where I learned if you have a lot of variables and they are, especially when they're highly correlated, it increases the chances that true associations are diluted and makes it a lot harder to separate the individual effects of those variables. So one way to address this is by creating correlation matrices, which allow us to see the correlations between certain sets of diagnoses. So as you can see, hopefully, there's a lot of blue in this correlation matrix, indicating that there are a lot of positive relationships between certain sets of diagnoses. And basically what this indicates is that when one diagnosis increases, the other diagnosis is also very likely to increase. And so because of these many correlations happening between these 19 diagnoses, it increases the likelihood that the individual strength of predictors is a little different in that complex model. But I think the biggest takeaway from that is, so this is the results from the multivariate analysis. You can see that there are a lot of statistically significant diagnoses for each of our psychosocial factors. But I think the takeaway is just that when doing predictive models, these models are fine, but it's also important to be aware of potential correlations in the data and to see what effect that they might have on the predictive model. So these are all very initial analyses based on just a couple weeks of R and statistics experience. But if I had 10 more weeks and more time, there are a lot of future directions to take this research in. A couple of these future directions are to identify specific damaging combinations of psychosocial factors and chronic conditions to see what adverse health outcomes they might cause and to see what populations they put at higher risk and why. But ultimately, the impact of this research is that it enhances our ability to identify vulnerable populations of vulnerable populations of patients and to tailor specific interventions to those populations to prevent poor health outcomes like disease exacerbation or hospitalization before they occur. So as I said at the beginning, I came in with a lot of experience in public health and very little experience in informatics, but am walking away with a broadened sense of understanding on informatics techniques and how they apply to the real world, as well as the implications for research like this. And while I have a better sense of appreciation for informatics and data analysis, I also learned a lot about the messiness of data and how hard it is sometimes to get it to do what you want. So in the process of working through R, I think I learned that the process a lot of times in data analysis is more important than the outcome, and that what we do in the process of cleaning, manipulating, analyzing our data shapes the reliability and the quality of our outcomes. And last, I learned a lot about navigating and utilizing resources available to me, whether that was the people at CMP or the countless R community forums out there. So now I'd just like to close by saying thank you to my mentor, Dave Dorr, and my manager, Christy Pizzamenti, and my colleague, Sumit Singh, for all of their support and guidance through my work on this project. And of course, to my wonderful Care Management Plus team for being the most fun and empowering group of people I could have worked with this summer. And then, of course, I'd like to thank the department, DMICE, and NLP training grant for making this internship possible for all of us.
Talk. So if you were going to collect some more data and you were specifically going to follow up on your substance use findings, looking at your correlation matrix, which of all of those multimorbidities would you focus on?
Oh, I wish I hadn't closed my laptop. Of finding the correlations? Yeah, well, I had a slide on the specific highly significant correlations and the coefficients. I think a really interesting one that we found was, so one of our diseases of interest was severe mental illness, and that seemed to have a lot of correlations for good reason with conditions like schizophrenia and dementia. So I think I would want to explore those first. Those seem to have the highest correlation coefficient in the correlation matrix, and so I think exploring that data would be interesting and to see the predictive value. I think also when I was just exploring data, I found a lot of information on homelessness and substance use, and I know from other research that's also a very highly significant relationship there. So I think that's one I would like to explore a little bit more also. Yeah. Oh, I still have this one.
Which is how to increase the manual curation for us by introduce the natural language processing technology.
Yes, so hello everyone. Thank you for coming to my presentation. Originally I planned this to be a 30-minute presentation, but it looks like I have less than 10 minutes now, so I'll try to get us all out of here by 12:30. But yes, I'm going to be presenting on how to integrate the natural language processing into our Reactome curation pipeline. So what is Reactome? Now, Reactome is an open source an open access pathway knowledge base. What that means is that in the past years, there have been an increasing amount of biological data being sent out by research, being published, and it's very hard to keep this all together. And so what Reactome does is we try to get all this information for pathways such as metabolism, signal transduction, gene regulation, and other biological processes, and package it together and make it easily displayable and easier to analyze for lots of different researchers. Each of the pathways that we're using is traced to primary literature, and we extensively cross-reference all of the bioinformatics databases with primary research and other professionals in the field. And we have tools also for browsing and visualizing the pathway data and analyzing the user data. And fun fact, when Dr. Wu was starting out on this project, I was entering kindergarten, and I'm presenting that. So why are we using Reactome? So there are other pathway databases such as KEGG and Wiki Pathways. And one of the advantages that Reactome has over these pathways is that it's open source. KEGG currently, it used to be open source, but now you need a license to download it. And as researchers, we all like free things. We have Wiki Pathways, which is open source, but it incorporates a wiki-style editing. So it's not as reliable. You could get some mistakes here and there. We have a very comprehensive overview of all the information that goes into Reactome, so you can be sure that our data is very accurate. So these are some of the advantages that Reactome has over some of the other pathway databases that are out there. And here are some statistics for Reactome usage. You see that on average for 2019 per month, we have 8.04 million monthly web requests and 75,000 average monthly users. And some peak numbers as well. You can see 9.74 million average web requests in March and 87,000 unique users in April of 2019. So you can see that Reactome is highly useful to the biological community by how much use it gets. So where does all of this data come from? Well, we have authors and we have curators that are used to write the modules and pathways. So we have a specific data model set up, and our curators go into it and actually write down the specific pathways and format the information so that it can be used by our program. Authors use our software tool to describe their pathways. All molecules must be identified and backed up by the primary literature. Reactome internal curators work with authors to ensure the consistency and completeness, and it's checked by multiple levels of peer review. As you can see on the right side of your screen, we have kind of like the pipeline of how this process goes down. So the editor-in-chief recruits experts and assigns curators. They work together on a module. That module gets okayed. Then it goes under a first step of review. Maybe revisions might be needed. Then it goes to an external review, and then that might be sent back for more review. So we have a very rigorous process of reviewing data before it comes into Reactome. Now, what are some of the problems that come with this? Well, this is a very slow process. We have to have curators go in and put in information, curators that are experts in their field. Right now, we have around 10 curators working on this globally, and every month or so, every quarter, we push out a new version of Pathway, of Reactome, which has more information, but it's a very slow process. And so part of my work for this summer was trying to use this tool called REACH to make the process more efficient. Now, what is REACH? REACH stands for Reading and Assembling Contextual and Holistic Mechanisms from Text. It is developed by the University of Arizona CLUE Lab, and it's a natural language processing tool, which essentially you can take a research paper, send it in, and it will output all the different pathways, all the different reactions that it finds within the paper, as well as contextual information and the participants for the pathway. So it's a very useful program for us because this is perfect. This is what our curators need to be able to add more information to Reactome. And so some of my project goals for this summer were to develop a user interface where curators could easily access these REACH-mined entities and use that to incorporate into Reactome. And then the second part, REACH has its own data model, which it outputs the information into. It outputs it into an XML file. But we have a specific model for Reactome. So we want to somehow be able to take those results that REACH outputs and turn it into our Reactome reaction-based data model. And I'm going to be focusing on this first option. So right now, we're not actually using the REACH program itself. We're using a server API that is developed by Chris Sanders' lab in the Dana-Farber Cancer Institute in Harvard. And what they've done for us is they've mined through more than 400,000 papers and sent it through REACH and got the output data from it. And they've developed an API for us to use. Now, an API, if you don't know, it's just it's a interface where you can send in information and then get some information back. So as you can see here, we're sending information to get the PubMed Commons ID, all these different qualifiers, and then from the output we can get context information, we can get what journal the paper has come from. So we're using this API for our project. We're not using REACH specifically. So here's the information flow diagram for how this is all going to be done. First, we're going to call the server using the specific queries that we want to get, the specific information that we want to get. It's going to run into my code, which is going to display it to the curator. We display it to the curator, and the curator will have an option to either accept or reject this to add this to Reactome. So hopefully that's the final place that we want it to get to. It's not at that point yet, but we're still working towards that. So starting with this internship, first I had to study all of the existing code, and this entire program is written completely in Java, so there's a lot to go through. This is all the packages. In Java, things are organized into packages and into classes, so we have all the packages just for the curator tool. This is not even for Reactome on its own, and all the different files in a single package, as you can see here. So there's a lot of information that I had to go through over this summer. It was a bit overwhelming when I first stepped into it, but that was part of just getting used to the workflow. Next, we have to decide on entry point. As you can see here, this is what our curator tool looks like right now. This is a specific view called the entity level view, where we see biological, we see proteins, we see ATP and ADP, we see where things are going on inside the cell. And where we're going to be able to use this is, say if you right-click on a protein, right here, RB1CC1, there will drop down an option that is search REACH. And from there, a table will be pulled up, the information will be sent into the REACH API, and then we'll pull all of this information out and then display it in this table as you can see here. So you can see there's the protein name, the protein ID, the specific PubMed Commons paper that is linked to, otherwise seen here as the PMC ID, and then a brief text which shows a little bit of context information on that specific reaction. And actually, we also have filtering options, so you can filter based on journal, you can filter based on year, and each one of these options here in this table is actually linked directly to the PubMed Commons paper that it comes from. So you could double-click on each of these results, and it would bring you straight to the web page. I was going to do a demonstration, but unfortunately, that didn't work out, so you have to look at this instead. This is the final product of what I've been working on for this summer. And so we have some future steps, which is to turn this information that we got here out of REACH and turn it into the data model that Reactome uses. And currently, we're studying, in order to do that, we're studying some programs. Right now, REACH, we have some programs that outputs REACH format and turns into biopacks, which is similar to our reaction-based data model. It's not exactly the same, so we're studying those programs to see how we can implement it into our program. And then otherwise, optimize the program for other entity types. Right now, we just have proteins, but we should try to look for other molecules, other different gene products. And lastly, I would like to just thank Dr. Wu and his lab for being so gracious and letting me work on this for this summer. I would like to thank the DMICE department, the Pathway Commons team, which is the larger team that works on Reactome as well as other projects, and also the National Library of Medicine grant for allowing me on here. Thank you very much. So, any questions? Thank you.